By the way, you may have to excuse any cat noises because one of the cats has climbed up in a strange part of the desk and is knocking things over. I will do my best to pause if that happens, so that way you don't hear me yelling at her. All right, so we have our first problem. The molar concentration of a sugar solution in an open beaker has been determined to be 0.3 molar. Calculate the solute potential at 27 degrees Celsius. If we're calculating a solute potential and we have a concentration, that means we are likely going to be using this formula. So let's see. We always have R because it is a constant. I am not going to go into any nitty-gritty details of the math, just to save time. If you are interested in learning some math tips and tricks, contact me for tutoring. So our ionization constant is important. What is our ionization constant going to be? Well, we need to look at what the solute is. It is a sugar. And we need to think, is sugar ionically bound or covalently bound? Well, sugars, those are all covalently bound. So any sugar has an I of 1 because when you dissolve a bunch of molecules of sugar in water, they're only going to make as many particles as you have molecules. Whereas if you were dissolving an ionic solution like sodium chloride, it would produce twice as many ions as the, quote, molecules that you put in. So our I is going to be 1. We need our C, which is our concentration, and they've lovelily given us that. And we need our temperature. Now, they gave it to us in degrees Celsius, but we're going to need that in Kelvin. So we're going to add 273 to that, and that's going to make it 300 degrees Kelvin. Looks like we've got everything we need. The only thing we have to do is plug and chug, so plug in our numbers and do our calculation. So our solute potential is going to be negative. Don't forget the negative sign. 1 times 0.3 times 0.0831 times 300. I know I've left the units out. That is to save time, to not have to go over unit math. My AP chemistry teacher would probably smack me on the head. He would be very unhappy with me right now, but we're saving time. So the calculation, when we stick it in a calculator, and like I said, cat noises, gives you negative 7.48 as our solute potential. I've heard different philosophies on whether you should box your answer if you have a math problem or not. I always box it because I have graded some papers where there's a whole bunch of math and I have no clue what the answer is supposed to be. And then you have to like dig through everything, and which makes it harder for the grader. The main problem is if you did a bunch of math and you put a box around the wrong part of it, that will be counted as your answer, and it's less likely that you'll get credit. All right, so we've got our solute potential. Let's do the second problem now. So we want the water potential. Let me go back to highlighting. We want the water potential of a solution of 0.15 molar sucrose in a beaker at 20 degrees Celsius. So a few things that we're getting from that. We have two equations that we're dealing with. We have our water potential is going to be our solute potential plus our pressure potential. So that's something to think about. And then we've also got the one we used on the last problem, where our solute potential is negative ICRT. So let's see what information we have. We have a concentration. Okay, our concentration is 0.15. We always know R, which is 0.0831. Our temperature is going to be 20 degrees plus 273. which means it's going to be 293. Do we know an I? Well, what type of molecule is a sucrose? Sucrose ends in OSE, which you should always remember means you're looking at a sugar. And all sugars have an ionization constant of 1. Okay, now we want the total water potential. For that, we need the solute potential and we need the pressure potential. They didn't give us the pressure potential. Well, they did, however, tell us that we've got a nice happy beaker with water in it. A beaker is an open container, and the pressure potential in an open container is always 0. So that means we can basically ignore this term. All we have to do is calculate our solute potential, and then we're going to have our answer. So let's go ahead and plug in some numbers to the second equation. So our solute potential is going to be negative 1 times 0.15 times 0.0831 times 293. And when you multiply that out with the calculator, if I did my calculations correct, you should get negative 3.65. And since we already established that the solute potential is equal to the total water potential, that's our water potential, that's our answer. By the way, if you are writing an answer and it starts with a decimal, please always put a 0 in front of it. And the reason for that is because if you just draw a decimal real quick, it may not look very close to a decimal, which means that you may end up getting points taken off because they basically can't read your answer. So be careful about that. If we do a all online FRQ only AP test, then the likelihood is that you won't have to do any math because it's a little harder for them for people to put that online. But I wouldn't put it past them. There's always the possibility that they'll surprise us with some math. So let's see. You measure the total water potential of a cell. So we got total water potential and find it to be negative 0.24 megapascals. If the pressure potential is 0.46 megapascals, what is the solute potential? So we want to know what is the solute potential. We are given the total pressure potential at negative 0.24, and then we're given the pressure potential, which is 0.46. Well, if we want to find that out, we got this handy dandy formula. So if we want to basically solve this formula for our solute potential, we can subtract the water potential or pressure potential from both sides, and that will give us our solute potential is going to be our total water potential minus our pressure potential, which if we plug in numbers is going to give us negative 0.24 minus 0.46, which is going to be negative 0.7. And when in doubt, try and put a unit down. So they gave us a unit for pressure. So let's use that unit just to be on the safe side. All right. So that is one of the easiest types of questions you can get. Pray for one of those. All right. A student calculates the water potential of a solution inside a dialysis bag at 22 degrees Celsius to be negative 6.25 bar, and the solution surrounding the bag to be negative 3.25 bars. What is the direction of water flow? And for this, it's a little helpful to do a diagram. So we've got no idea what the solute or anything else potentials are. We just know that we've got a bag and it's sitting in a solution. And I know it looks kind of like a candy, but that's actually kind of what the dialysis tubing looks like. All right, so our water potential, which I'm going to call psi d for dialysis tubing, is equal to negative 6.25. Our water potential of the solution is negative 3.25. And I'm going to call that psi s for solution. Honestly, it doesn't matter what you call it, you just need to differentiate. And actually, this question does not require you to do any calculations. It's a little more conceptual. So you can guess the direction of water flow if you know a couple of things. If you know the tonicity, or if you know the water potentials. Because water moves from a high water potential to a low water potential. So we just need to figure out which one of those two numbers is lower. But here's the thing. You might be automatically deciding, oh, negative 3.25 is lower. But you forget we're dealing with negative numbers, and in negative numbers, things are a little different. So let's look at our old friend, a number line. And let's say that this is 0. Everything to the left of that is going to be negative. So this is negative 1, 2, 3, 4, 5, 6. So negative 6, negative 3. Well, our solution has a solute potential around 3.25, and our dialysis bag has a water potential of basically negative 6.25. Anything that goes to the left is a lower number. So what is the smaller number? Well, the smaller number is that dialysis bag water potential. So what that means is negative 3.25 is greater than negative 6.25. Kind of backwards when you're dealing with negative numbers. So keep that in mind. Nobody's going to judge you if you have to sit there and draw a number line to help you remember this on the test. Honestly, I'll be sitting there going, good, you learned. All right. So what that means is that the dialysis tubing bag has a higher water potential than the solution outside. What that means is water is going to move from the dialysis bag to the surrounding solution. And this is your actual answer, because you're describing what is the direction of the net water flow. All right. And last but not least, we are given the following, not what I meant to do. Excusez-moi. All right. If a plant's pressure potential is 2 bars and its solute potential is negative 3.5 bars, what is the resulting water potential? This is a nice, easy one to end on because we've got our pressure potential is 2 bars. Our solute potential is negative 3.5 bars. And we're looking for our total. Well, we happen to have an equation that will give us this lovely information. Just straight up give it to us. So it's going to be 2 bars plus negative 3.5 bars, which is going to give you a total water potential of negative 1.5 bars. And there is your answer. Ta-da! Easy peasy. All right, so let's talk about pressure potential real quick. It's also called turgor pressure, and it's actually very important when you're talking about plant cells. So, plant cells have something different from animal cells. They have a cell wall surrounding them. And when they're surrounded by a cell wall, that helps keep a cell from lysing. So, what's going to happen is with a plant cell, if you stick it in a hypotonic solution, water is going to enter this cell. Water is going to enter this cell until basically the cell wall keeps this thing from expanding. So there is a limit to how much water can enter the cell. Basically, things are going to move until the pressure, until the total water potential is equal on the inside of the cell and the outside of the cell. So what's going to end up happening is you're going to have some solute potential, and then you are going to have an equal but opposite pressure potential, basically provided by the cell wall pushing in. And that's kind of putting pressure on the solution, which is going to kind of help you out because if you're calculating the total water potential, if there's some pressure potential pushing in, that's going to affect how water will be able to move. So this is generally measuring the forces of the cell wall pushing inward. As more water enters the cell, that pressure increases. And eventually, it will increase until basically the pressure potential and the solute potential are equal. That's if the cell is not doing anything to actively disrupt this. Just naturally, that is what would happen. So, turgor pressure and wall pressure are basically equal and opposite. So, you've got both of these increased by water entering the cell, and turgor pressure is counteracted by wall pressure, which is equal and opposite. So, whenever turgor pressure increases, the pressure of the cell wall is going to increase as well, pointing against it. Basically, you'll reach a state of equilibrium where your cell is bulging full of water, but it is not capable of taking in anymore. And like I said, this will ultimately end with this thing in equilibrium. So let's say you put a potato cell inside of a beaker of water. All right, it's pure water, got no solute, it's in an open container, got no pressure potential. So the total water potential of this solution is 0. Same thing in either case, because we're saying no solutes moving. Well, this potato actually has some total water potential. What's going to happen is it's got no pressure potential when it's put in, but it's got solute potential because there is water. Well, because there's stuff inside. So the potato cell has a lower potential, and the pure water has a higher potential because 0 is bigger than any negative number. This is the total potential of the potato cell. So that means that water is going to go from the higher to the lower water potential, which means it's going to enter the cell. When it enters the cell, it's going to keep entering until basically the cell is pretty much bulging full of water, and it's going to keep entering until it's so full that it's basically causing turgor pressure against the cell wall, and the cell wall has to exert an equal and opposite pressure against it. So that way the system will be in equilibrium, and all the water movement that we have is going to be, equal. Whatever comes in goes out. So let's draw some equilibrium arrows. Okay. So, what's important about this for plants is that based on the different water potentials in different parts of the plant, that actually drives the movement of water. So we talked about cohesion and adhesion and how that helps a plant to transport water up through its vessels, so xylem and phloem. However, just adhesion and cohesion alone are not enough to fully counteract gravity. Instead, there are water potential differences that are going to help this water move. So basically, as you go higher up in a plant, let's say you're looking at a tree, you're going to get lower and lower water potential. Remember, water moves from a high water potential to a low water potential. The soil, has the highest water potential. The atmosphere has the lowest water potential. And so, um, basically, the plant has it set so that its root water potential is just a tad lower than the soil. So water will naturally move from the soil into the root. The plant doesn't have to expend any energy. All it needs to do is change up the solute concentration inside its root cells. It can do that by adding in some glucose or taking a big molecule and breaking it into smaller chunks. Then the stem has a slightly lower water potential than the root, which drives water to move from the roots up to the stems. Similar thing with the leaf. The leaf has a lower water potential than the stem, and so water is going to naturally move from the stem to the leaf. And then the atmosphere has the lowest water potential of all, so water is naturally going to move from the leaf to the atmosphere. So if we see this in action, what we see is in this diagram, we've got our soil water potential is negative 0.3. It decreases when you get into the xylem trunk. As you go higher, it keeps decreasing. Because remember, when we're looking at negative numbers, the bigger the number, when it goes from like 1 to 7 to 100, that means it's actually a smaller integer. So outside has the lowest water potential, which means water is going to be driven to the outside atmosphere. How does a plant keep from losing all of its water before it gets to the leaves? Well, the xylem, which is basically where water is being transported, it doesn't have any holes in it. The only holes that water can escape out of to get to the outside air are in the leaves. So water is forced up into the leaves, and then it will escape into the atmosphere, which has the lowest water potential. That's pretty cool. And if a plant, say a plant has, there's a lot of transpiration going on, so there's a lot of water evaporating from these leaves, it can adjust its water potential of the different cells in order to continue to drive water up. Why is that important? Because plants don't have a circulatory system, and that water that's in the xylem carries nutrients and other stuff dissolved in it. So if you don't have that xylem flowing up, you're not getting the nutrients you need up to the top of the tree. All right, now let's look at osmoregulation. Basically, we looked at how basically the osmosis affects the function of a tree. Now we're going to look at what happens in animals. So you and your cells are constantly exchanging material between the interior of your cell or your intracellular space and the exterior of your cell or the extracellular space. And what you pretty much do is your cells can either use facilitated diffusion, so some carrier proteins, or they can spend a little energy by active transport to adjust solute concentrations so that water will flow in the appropriate direction. This happens in your kidneys. This happens, with relation to your blood, and this helps maintain homeostasis. When this goes wrong, bad things can build up inside the cell, like toxic waste. Not toxic waste like nuclear waste, but toxic waste products. And also you can have excess water buildup. And remember, our cells don't have cell walls. If we get too much water in our cells, they're going to pop. They're going to lyse. So mammals have these mechanisms to regulate our osmotic pressure, and we tend to concentrate on 3 major types of fluid: your blood plasma, your extracellular fluid, and your intracellular fluid. And so your body, your cells can regulate the osmotic pressure of each of these by adding in electrolytes, which are basically ions, or taking out electrolytes. And by that, you can basically change up the osmotic pressure. So that way you'll have, the flow of materials or the flow of water into the cell, into the intracellular fluid, or if it needs to exit the cell, going out to the extracellular fluid. So here is a diagram from an AP test question. And what I want to focus on is I want to focus on the open flower. So in the open flower, you have a potassium channel letting potassium in, and it's increasing your concentration of potassium. So it's building up potassium in here. All right. Then you have a proton pump, which is actively moving hydrogen into this vacuole. And then you have a transport protein. It's a sodium hydrogen transport protein, and it's going to use active transport, so it's going to use up some ATP. And what it does is it actively exchanges potassium and hydrogen. So it's basically, you're shoving a bunch of hydrogen into this vacuole, and you're using that hydrogen that's in there to add in more potassium. What this does is it increases the solute concentration, which, if you remember, is going to decrease our solute potential and therefore our water potential. And so you have a higher water potential outside, or even outside of the vacuole, but still inside the cell. Water always moves from a high water potential to a low water potential, and that's what's driving the movement of water into this. And that movement of water is going to go into the cell and fill up that vacuole and helps that cell expand in size. So by moving around different ions, the plant can manipulate how things move. And honestly, for a lot of plants, stuff like this is one of the major ways they can move. They either grow towards or away from something, or they can kind of swell up different parts of their body with water. So, if you are a fish, you've got different problems. So, in the top, we have a fish living in a freshwater environment. That means that this is all hypotonic. And the interior of the fish is going to be hypertonic. And water always moves from hypo to hyper. So notice that you've got a bunch of water will basically diffuse into this fish. But remember, we're animals. We don't have cell walls. So if we don't have a cell wall to prevent our cells from lysing, we need to get rid of that extra water somehow. So the fish has a couple of adaptations to make sure this happens. One, it excretes very dilute urine, which is going to release a lot of water. It's also going to actively take up ions through its gills. And so actively taking up those ions is going to help change the water potential. And then, so you're also going to have this fish drinking very little water. So it's trying to take in as little water as possible and remove as much as possible because water comes in through the skin. Then we have a saltwater fish, and it's going to have slightly different adaptations. So we have a hypertonic environment, and we have now a hypotonic fish. That means that naturally, water is going to exit the fish. It's going to diffuse out, and so the fish is going to lose water through its skin. Because of that, if that keeps happening, the cells will shrivel up and no longer be able to work. So this fish drinks a lot of water. When it drinks a lot of water, it's going to release extra ions through its gills, and that is going to allow it to retain the water, and then it excretes a very concentrated urine. So notice the size of the arrow, there's a lot less urine coming out, and it's going to be a lot smaller in volume. Here is a paramecium. So paramecia live in fresh water. So they are living in a hypotonic environment, which means water is going to naturally rush in. Paramecia, I forget if they have cell walls, but even if they don't, they need to be able to somehow regulate their osmotic pressure and make sure they don't have too much water rushing in. So here's what happens. They have something called a contractile vacuole. It contracts, and then what happens is water gets taken up through these canals. You don't need to know the parts of this. You just need to know that this contractile vacuole starts filling up with water. When it's full, some pores open up to the surrounding, and the vacuole is going to contract. It's going to actively squeeze out that water, and then it's going to be empty and ready to take up excess water again. So, as long as this paramecium is in fresh water, it will be able to actively expel water that's coming in. This I'm only gonna like very briefly go over. I just want to note that you have active and passive transport going through. You also have certain areas of this that are water impermeable. And what I want to point out is that this is part of the tubule system in your kidneys. So, you've got measurements of osmolarity and so on. But what I want to point out is that here you're actively pumping salt out into the surrounding, but this area is impermeable to water, so it probably doesn't have any aquaporins or anything. That means that you're going to concentrate the solution that's outside of this tubule. Inside this tubule, you've got a solution that, has salt in it, or has stuff in it. It's probably not going to be permeable to sodium chloride. However, it is permeable to water. And so since you have this concentrated solution outside, that drives water out because water is going to move from the hypo to the hyper. Okay? That is going to result down here at this nephron loop in a very concentrated urine. So it's highly concentrated. Then, once it comes up here, that is when you're going to start pumping out sodium chloride, which is going to dilute the urine. You're keeping the water, but you're diluting the urine. And that is basically going to be controlled by your body. If you are not drinking enough water, you will probably have either less leakiness of your sorry, I don't remember the parts of this, of the tube that's going down versus the tube that's going up. And if you are drinking a lot of water, well, then you might have this might be kind of leakier to let more water, actually not, it'll let more sodium out and it will allow water to go through. Basically, your urine is very concentrated if you're not drinking enough. So it's less water with a lot of solute mixed in, or the same amount of solute mixed in, which is just your waste products, like urea and a few other ions. And then if you are drinking a lot of water, you're going to have very dilute urine because you're going to be leaving a lot of water in it with the same amount of waste, etc. All right, so this is also looking at the same thing, except this is looking at it on a cell level. I'm gonna not go over this in detail, but you can pause this video at any point, and I would sit here and read all of this and look at kind of what's happening if you have time. It's cool because it shows you ion channels, aquaporins, and transport proteins. Remember, a transport protein is always going to use active transport. Ion channels can be passive, and aquaporins are also passive. So you use energy basically to pump solute into these cells that are lining this tubule. And then you're basically going to be exchanging sodium and potassium. So that's a sodium-potassium pump. Then you're going to have sodium leaking out through this ion channel, and it is going to increase the concentration out here. Some of that will rush into the blood, and basically water follows solute. So if you start moving solute from the urine to the blood, water's going to follow that. And then this is just another point. When you have movement of stuff across the membrane, that is going to allow, and more cat noises. That is going to allow growth of the organism because you're taking in new molecules, and that's what you use to build the biomolecules that are making up your cells. So that is fairly simple. That is the end of that. I hope you enjoy this watery picture that I got from that website, where this dude apparently has a bunch of different photos. Next up will be Unit 3.